Okay, so now we're going to talk a little bit about the role of probability in genetics. During a cross, uh, an event wherein the gametes of two individuals come together to make another individual, there are a lot of things that go on that are kind of dependent upon chance. The things that are dependent upon chance are the gametes. Each gamete, there's a certain probability that it's going to inherit certain alleles from the parent, depending upon how the genes, how the alleles segregated or independently assorted during meiosis, and the probability that two gametes are actually going to come together and meet each other, and that involves in plants like pollen tubes growing down into the ovaries of plants, and in animals the sperm swimming up to meet the egg. So you can think of some of these phenomena as sort of like flipping a coin. For instance, the segregation of alleles into eggs or ovules is kind of like flipping a coin, wherein heads is 50, an outcome that you expect 50% of the time, and tails is an outcome you expect 50% of the time. And so you can think of heads as big P and tails as little p. And same thing for the generation of sperm. Now, probability varies between 0 and 1, with 1 being completely certain and 0 being completely improbable. In a coin toss, for instance, you have 1/2 heads, 1/2 tails, 50% probability that's going to come up heads, 50% tails. In a die roll, you have fractions, proportions, probabilities, percentages of things like 1 out of 6. Now, we're going to investigate something now called the rule of multiplication. This is the idea that how to calculate the probability of two events happening. So what's the chance that two coins will be tossed will both be heads? And the way to determine this is that you compute the probability for each event and then you multiply those together. And so the probabilities go down. And that kind of makes sense. If there's a 1/2 probability, a 1/2 chance that the first coin you flip is going to be heads, 1/2 chance the second coin that you flip is going to be heads, multiply them together, and the 1/4 chance that the two coins that you flip are going to be heads. This kind of makes sense because there's really 4 outcomes, right? There's 2 heads, a head and a tail and a head, and 2 tails. So 1 out of 4 sort of seems intuitively right, which is important. Okay, now, and this sort of reminds us of the Punnett square when, if we look at, and let me choose this magnifying glass here, we look at the case wherein what's the probability that the two alleles of an offspring of two gametes coming together are going to be both dominant, both big P's? Well, that's kind of like the probability that two coins flipped are going to be heads, so that's 1/4. What's the probability that you're going to get this particular genotype down here at the bottom? Homozygous recessive. Well, it's sort of like flipping a coin and getting 2 tails. So you can see how probability can be used to describe what's going on here. So to apply it to monohybrid crosses, the probability of receiving a homozygous recessive when 2 heterozygotes mate, you calculate the probability that each of the heterozygotes produces a big P gamete and a little P gamete. Each of those are half, and so segregation is in a sense like flipping a coin. Now, in dihybrid crosses, you calculate the probability that 2 heterozygous individuals will produce, let's say, a double homozygous dominant offspring. So it's the probability that the heterozygous, you calculate the probability that each heterozygous individual will produce a gamete, which is, big Y, big R. And that is 1/4. And then multiplied by 1/4 is 1/16. That kind of makes sense. 1 out of 4 gametes have to be in a certain configuration, and the other gamete has to be similar, and so bring those together, and that's 1/16. Okay, now let's examine the rule of addition. This is calculating that the probability that, either of two results could occur instead of, two results occurring one after the other. So it kind of makes sense that this, well, you add the individual probabilities together, and probability gets larger. So this is like calculating the probability that you're going to get a heterozygous individual from crossing two heterozygotes, because there are actually two outcomes that can occur to sort of fulfill this, either this one here or this one here. And how you calculate the probability of any heterozygote being formed in the next generation is you calculate the individual probabilities, 1/4 and 1/4, and you add them together to get a probability of 1/2. Now, the value of these rules of multiplication and addition are that they permit fast calculations of expected genotype and phenotype frequencies, and they're faster than filling out Punnett squares. If you are doing trihybrid crosses, tetrahybrid crosses, in addition to monohybrid and dihybrid crosses, you could see how the rules of probability would simplify those calculations.